welcome you to this pre-congress workshop on clinical research methodology. Without any delay, because we have already 8 minutes delay, I would like to request our chairperson, Professor Eminent to say a few words and start the session. Professor Eminent.
It's a great occasion for us for the Oncology Club. We have organized with the help of Tata Memorial Hospital, clinical research methodology, a workshop on clinical methodology and protocol.
I would like to request everyone to please mute yourself, otherwise it will be a problem to conduct the sessions.
We are trying for a pretty long time to award some money for research protocols. We couldn't get any research protocols. That's why we took the decision that we shall have to train them how to take research protocol and how to do it. That is why we're giving you trouble. From Goda and Bikram making problem. Dr. Jain Goda and Bikram Goda. Jain Goda and Bikram Goda. Thank you all very much. Hopefully our workshop will be a good one. After the completion of the workshop, we will be trying to develop a protocol. I will certainly try to finance them. Thank you all very much. I'm not going to take any time. We are very late. Over to you, Dr. Jan.
Thank you very much, Professor Hai. Thank you, Dr. Kamaluddin, and thanks a lot, Dr. Suman, for inviting us to conduct this session. Vikram, my dear friend, I don't think he requires any introduction. For he has been spearheading lots of clinical research methodology workshop here at ACTEC and includes associated that of Accord. I'm pretty sure you'll like the way he's going to take his lecture, and please don't shy away from asking questions, because you've got the best person who is going to take this lecture in clinical research methodology. As far as this workshop is, try to keep it very simple. We are going to talk mostly about in these 3 sessions, which will be done on 12th, 19th, and on 29th. We are going to talk about simple retrospective studies, observational studies, and the statistical methodologies for those studies. Before we come to Dhaka on 16th, to give you a practical demonstration so that you are able to complete your concept development worksheet. So thank you very much once again. I hope you will enjoy the lecture given by Dr. Vikram. Thanks a lot, Suman.
So thank you, sir. Probably you have all received a mail just few hours later before. So if you need any kind of assistance, there will be 2 course coordinator. Probably you know them, Dr. Tawhidul Islam and Dr. Rifadara. You can contact with them. And in case any kind of problem, you can directly contact with our supervisors or our faculties. So Professor Vikram, sir, now the floor is yours. So I would like to request you to start the sessions. Professor Vikram, sir.
Okay. Yeah, thank you very much for in fact organizing this workshop in the first place. I think even in this part of the world, in India also, these workshops are kind of organized quite rampantly, because there is so much need to develop capacity in clinical research, and we are really too happy to kind of help students in Bangladesh develop capacity in this field. And so I would like to thank Dr. Mustafa, Dr. Kamaluddin for giving this opportunity, and also my friend Dr. Jain Goda, who kind of believed in me and thought that I'm the right person for this job. So with this, I will start my presentation, but I have a small request. Actually, it so happened that I'm not able to log in using the Zoom link from my laptop. So I have joined from my phone, but obviously I cannot present from my phone. So therefore, I've emailed my presentation to Dr. Mustafa, would it be possible for Dr. Mustafa to play the presentation so that I can talk from here? Is that okay?
It is okay. It is possible. Please send it by mail.
I emailed you. So yeah, you may put it in full screen mode.
Okay. Yes, sir. My screen is visible right now.
Okay. Yeah, very much. So are the students able to see the screen?
Yes, sir. I can see the slides. Hopefully, everyone can see.
Okay. All right. Perfect. So good afternoon, everyone. As it is shown on the slide, my name is Vikram Gota. I am professor of clinical pharmacology at ACTREC, Tata Memorial Cancer, Mumbai. And I'm going to talk to you about clinical research methods and also very briefly about retrospective studies. So there's a very famous saying, next slide, please. There's a very famous saying. Can you go to the next slide, please? Yeah. So what it means is, research is a matter of finding things. What you don't know, you have to do research to find things. Next slide, please. Okay. So now, before we do research, there is something called good clinical practice that all of us should know, because good clinical practice encompasses two important aspects of any research. One is having good data integrity and having safety of patients in mind. These are the two important pillars of good clinical practice. And if you are not aware of GCP or good clinical practice, also known as the good research practice, then you are going to make a number of mistakes. And at the end of it, the clinical trial findings or clinical study findings that you have may not be actually reliable. So therefore, first and foremost, if you are not familiar with good research practices or whatever, there are various monographs and there are various documents which are available on good clinical practice. I encourage all of you to read that because it will tell you about the significance of consenting patients, and then the importance of having IRBs, and then getting IRB approvals, following the protocol the way it is written, and what should you do if there are any deviations, how should you take care of the patient safety, what should you do if there is an adverse event, whom it should be reported to, and how do you, so all those things, and how do you document whatever you are doing. So documentation is a very important aspect of clinical research. So all these aspects are given in good clinical practice guidelines. So these are guidelines and all of us should be aware of it. Next slide, please. Okay. So before you do a question, any research, why do we do research? When you're thinking about clinical research, there are 4 important aspects that you should remember. Okay. The 4 important aspects of any clinical research are: one is population. Okay. Whom are we going to do the study on? Obviously, if it is a diabetes study, you cannot be doing it in heart failure patients. Okay. You should do it in patients with diabetes. And if it is a type 1 study for type 1 diabetes, you cannot be taking patients with type 2 diabetes. So therefore, you have to first define the population. And then what is the intervention to be tested? So that becomes very important. So whether you're going to have some behavior as an intervention, or some diet as an intervention, or a medicine as an intervention, or an exercise regime as an intervention. So whatever it is, radiotherapy as an intervention, chemotherapy as an intervention. So whatever it is, so that should be clearly defined. Okay. So then, why are we doing research? Because we always continuously want to improve the outcomes of patients. So therefore, every time you do a research, take up a research study, it is quite possible that there will be a standard of care already available. So then what is your comparison strategy? What are you comparing your intervention with? So if you are coming up with a new intervention, let's say a new drug, so what are you comparing? What is your standard of care? Okay. Like for example, if I'm having a new chemotherapy for breast cancer, let us say there is a new HER2 blocker which has come for breast cancer, then obviously our comparator will become Herceptin. because herceptin is already used as a HER2 blocker in breast cancer patients. So that is the comparison. And then the final, this one, O stands for outcome. So what is the outcome of your interest? Like, for example, if you're doing, let's say, a case control study, whether somebody developed cancer or not. If that is your outcome, that you should specify that. Or how long is a patient going to survive with the strategy? Then your outcome of interest will be death. Okay, and for how long the patient will remain without progression with a particular treatment, then your outcome of interest will be disease progression. So these are the 4 important things that you have to define whenever you're going to do a clinical study. Okay, next slide. Now, the type of next slide. Okay, so the type of question actually determines the design. So basically, you cannot say that, okay, I know this case control study designs very well. So whatever question is there, I'll try to answer it through a case control study. No, it doesn't work like that. Actually, it's always the question that should dictate the design. Like, for example, if I want to say whether I know jogging every day for 5 kilometers is going to improve my cardiovascular event-free survival, then you should probably do some kind of a cohort study, okay, to understand that, or you can do a case control study. But on the other hand, if I have an idea that whether a drug is going to be effective for treating hypertension, then you are likely to have a prospective randomized controlled clinical trial, which is more which is best suited to answer your question. So that way, it is always the question that you have that should drive the design, and it should not be the other way around. Okay, and in the following slides, I'm going to talk to you about the designs and when to apply each one of those designs for what type of questions they're applicable, and so on and so forth. Next slide. Next slide. Can you please change the slide? Yeah. Okay. So now, broadly speaking, okay, what are the types of trial designs that we know of? Very broadly speaking, there are 2 types of trial designs. One is called as an observational study, and the other one is called an experimental study. In an observational study, you don't do anything. You will allow things to happen the way they are happening. Okay, you will just observe and you will document. Okay, so what will happen if I eat, let's say 10 chapatis every day? Will I become obese? So then you may pick up, find people who are eating that kind of, having that kind of exposure. You just follow them up, okay, and then see whether they become obese or not. Okay, so that is kind of some kind of an observational design. So where you are not going to intervene. So what you must understand is when you're doing an observational study, you are going to enroll patients in their natural behavior. Okay. So if that is the question that, if I eat 10 chapatis, whether I become obese, then you're going to enroll patients who are kind of eating that kind of, having that kind of a dietary habit, and then just observe them. So that is observational study. There is no intervention there. So you won't give them any statins or whatever, or you won't advise them to kind of go for walks and things like that. If they're going for walks on their own, that is fine. But you yourself per se do not observe. So your only interest is those chapatis and cardiovascular and obesity. Okay. On the other hand, experimental designs are like there is a treatment involved. Okay. The treatment could be once again anything. So whenever we say the experimental designs, we should not only think of either chemotherapy or radiotherapy or surgery or whatever. The intervention can be of any type. It can just be a behavioral intervention. Okay. Asking the patient to kind of, go for walks, or asking the patient to do yoga, or asking the patient to control his anger. Depending on what kind of question you have kind of designed, the intervention, the question you have in mind, the intervention has to be tailored to that question. Okay. So it is not always drugs. Okay. So when you come to, okay, now we understood that there are 2 types of study design, observational and experimental. Next slide. Okay. So in the next slide. Yeah. Okay. In the observational studies, there are 3 primarily, there are several types, but there are primarily 3 important observational studies. So one of them is called cross-sectional study. The other one is called case control study, and the third one is called a cohort study. Now, when do you want to do a cross-sectional study? Cross-sectional study is very simple. Like, for example, so you have, let us say, in this group, I can see that there are about 40 individuals. So now out of these 40 individuals, if I want to say how many are males and how many are females, that is a cross-section study. Okay, at this point in time, in this hall, in this meeting hall, let us say, how many boys and how many males and how many females are there? That's a cross-section study. In the city of Mumbai, what is the prevalence of diabetes? If I want to know, so I just do a cross-section study and then say, okay, and then 20% of people in the city of Mumbai are diabetic. So basically, these kind of studies are used to describe the prevalence of certain variables, and they're very easy to do. Why? Because it is a snapshot. It is like taking a photograph, like using a camera. Check. Okay. So at this point in time, you just do, okay, I counted 3,000 people and I found so many diabetics. So I think the prevalence of diabetes in the city of Mumbai is going to be this much. Okay. So that is called as a cross-sectional study. The strength of a cross-sectional study is it is very fast and it is cheap. It is not very expensive because you don't have to do any follow-ups, you don't have to do any intervention or whatever. But on the other hand, there is something called as cohort studies, which requires a little bit of a follow-up. Like for example, generally, if you have questions like, if I have certain habit, okay, what are the outcomes going to be? Okay, so those are the typical questions that are answered in a cohort study. Okay, so a cohort study can be prospective or retrospective. Okay, yeah. So if it is a retrospective cohort study, it is generally called as a cross-section case control study. So so the only difference between a retrospective study and the prospective study is, in a retrospective study, the outcomes of your interest should have already happened. Okay, somebody would have already had an MI. Okay, and then you go back and see between patients who had MI and patients who did not have MI, what is the what is the prevalence of cigarette smoking in those two groups? So basically, what your case control study will answer, the purpose which is given in the third bullet point here. No, I think are we on the same page? No, one second. I think there's some confusion. Types, strength. Okay, you're in the okay. So one second. Cross-sectional. Yeah. After cross-sectional, can you please go to the next slide? Can you go to the next slide, please? Yeah. So cohort study can be of two types. It can be a prospective cohort, wherein you identify patients who are having certain exposure, who are not having certain exposure, and you follow them up and then see whether they will develop an outcome of interest or not. Or it can be retrospective. In a retrospective study, what will happen is you will identify patients with certain disease and without certain disease, go back in time and then see how many people with disease had the risk factor and how many people without the disease had the risk factor. Go to the next slide. I'll explain it more closely in the next slide, please. Next slide. Okay. So the retrospective observation study is also called as the case control study, typically. Okay, wherein, as I said, there will be cases and controls. So cases are generally people with disease and controls are generally people without disease. Just to give you an example, you can have cases like diabetics, non-diabetics. And then you can go back and see what is the, whether the diabetics used to have more than 20 grams of sugar intake per day. Okay, whether having sugar intake more than 20 grams per day can make somebody diabetes. If that is your question, you can have diabetics and non-diabetics. Go back and see what is their sugar intake per day. And here you'll actually be, identifying what is the prevalence of that exposure. So in this case, the exposure is defined as somebody who is having more than 20 grams of sugar, who is eating more than 20 grams of sugar. That is the exposure. So you'll go back and see how many of what is the prevalence of that exposure in the diabetes group versus non-diabetes group. And if you see, that the prevalence of high sugar intake is more in the diabetes group, then you would have proven your hypothesis. On the other hand, if you see that there is no difference in the amount of sugar that this group and the two groups were eating, then you will disprove your hypothesis and say that, okay, there is no connection between sugar intake and diabetes. Okay, so that is a typical example of a case control study. Next slide, please. Next slide. Okay. Okay. So, as I said, these studies can actually evaluate the effects of an intervention. Once again, what is happening? I don't know. I think there is some confusion here. Okay. So anyway, so we completed the observational studies. We'll go to the experimental one. Yeah, we'll stay here. So I think you understood the observational studies, no? So observational studies, there are 3 types. One is cross-sectional studies, where you just take a snapshot. Case control study, where you take patients with cases and controls, go back in time, and then see what was the prevalence of an incriminating factor, okay, or an exposure. And in cohort study, you take people who are exposed and who are not exposed, and you follow them up, and then see how many of them will develop the outcome of interest. So case control is respective, cohort study is prospective, and a cross-section study is a snapshot that at that point in time. Now, after having completed that, we'll go to the experimental studies. Okay, so the experimental studies will help us to evaluate the effects of an intervention. And I said the intervention can be anything, like it could be behavior, it could be drug, it could be device, or whatever. Next slide. Okay, next slide. And all of us know. Next slide, please.
Next slide is there, Dr. Vikram. We can see the slide.
Yeah. I wanted this slide actually. Yeah. So now there are okay. So there are 4 phases of clinical trials: phase 1, phase 2, phase 3, and phase 4. And no matter what the intervention is, all those interventions can actually go through these 4 phases of clinical trials. In the phase 1 clinical trial, what we do is we generally try to see the feasibility. By feasibility, what I mean is, suppose you have a drug. Okay, so can you give the drug at all? Is it going to be tolerated? Okay, that is the feasibility. Or up to what dose can you give the drug? So I start with 5 milligram, go on escalating from 5 to 10, 10 to 20, 20 to 50 to 100. And then I see that at 100 mg, there is toxicity. So that kind of a study where you test the feasibility of administering anything or an intervention, administering an intervention, is called as a phase 1 clinical trial. Okay. And phase 2 clinical trial are generally therapeutic exploratory trials. Like, for example, if I'm using this drug, response rates. So if I'm giving a drug, what is the response rate? Like, for example, if your standard of care has a response rate of 50%, whether the new treatment can improve the response rate by 20%. So that is tested in a phase 2 clinical trial. Can you please go to the next slide? Phase 3 clinical trial. So in a phase 3 clinical trial, these are therapeutic confirmatory clinical trials. So once again, I'll repeat, phase 1 is feasibility assessment, phase 2 is exploratory, and phase 3 is confirmatory. In a phase 3 confirmatory trial, you will actually determine whether the treatment has any efficacy or not. So typically, you will have outcomes like progression-free survival, overall survival, to look for efficacy of the drug. And phase 4 is generally called post-marketing trials, because whenever you do a clinical trial, the maximum number of patients that you are going to test in a clinical trial is about, let's say, 2,000 or 3,000 or whatever. That's the maximum number of patients. But some of the rare adverse events, like, for example, adverse event that happens in 0.5% of cases or whatever, they may be missed, or you may not be able to document them very clearly, even in a 2,000 patient huge study. So therefore, under those circumstances, what the regulators will say is, okay, I will give you approval because you have shown efficacy data in a phase 3 clinical trial, but go to a phase 4 trial and demonstrate when the patients are actually taking the drug in a real life situation, in a real world scenario, you demonstrate the safety of the drug. So those kind of studies which are done after the drug receives marketing approval are generally called as phase 4 trials or post-marketing studies. Next slide. Next slide. Okay. So now, just to understand, so we kind of understood what are the different phases of clinical trials. But now, what is a clinical trial? Okay. So a clinical trial is something which is done to test the efficacy of a drug, to identify for various outcomes, like, the outcomes such as you want to study the safety, you want to define the dose of administration, you want to test a new drug formulation, you want to test, combining two treatments together, or you want to even look for quality of life of a new intervention. So any of these things can be addressed with the help of a clinical trial. Next slide. Next slide. So now, hitherto, what we looked at is, we looked at the phases of development. Phase 1, phase 2, phase 3, phase 4. They are not actually the types of clinical trials. They are phases of development. So phase 1, you will just see whether it is feasible. Phase 2, you will see whether it is, you will explore its activity. Phase 3 will explore its efficacy. And then phase 4, you do post-marketing surveillance. But they are not the types of trials. The types of trials are something like whether you are doing an intervention to treat a disease, or whether you're doing an intervention to prevent a certain condition, or whether you're doing an intervention to kind of early detection, like, for example, somebody when they developed, let's say, pap smear, okay? So that was a screening study, okay? So, and then you have diagnostic trials, like, for example, how good is a new intervention over the existing one for diagnosing a disease? Like, for example, you may want to compare, I mean, when CT scans came into existence, till then, the best diagnostic tool would have been, let us say, X-ray. So then you do a comparison between CT scan and X-ray, and then see which of the two is better at picking up a disease. So these are the different types of indications for which you can do a clinical trial. Okay, next slide. And when you look at various phases of development, you typically, when you look at the numbers, a phase 1 trial will typically have about 15 to 30 patients, okay? And they're generally first in humans. And phase 2 is typically about 100 patients. Phase 3 will have, a few hundred to few thousand patients. And here you compare your new agent with the standard agent. And phase 4, as I said, so therefore it's called as a confirmatory clinical trial. And phase 4, as I said, is to find out the long-term safety effects. Next slide. And who are the players of? Okay. So now, who are the players involved in a clinical study or in clinical research? In clinical research, you definitely, need human, human patients or volunteers, depending on what kind of a study is that. You need a doctor who will administer the treatment. You need research nurses to carry out the orders. You need pharmacists to kind of dispense the drug and deliver them. And you need lab technicians to do analysis. You need social workers to sometimes counsel the patient, and you need data managers to manage the data. So if you have all these ingredients, you will have the set of people who will take care of the safety of the patient, and a set of people who will ensure that the data integrity is maintained. So those are the two important pillars of good clinical practice that I mentioned in my first slide. Next slide. Okay. Next slide. Okay. So now, how do you start doing a clinical trial? Okay. So the best way to start doing a clinical trial is to have a written protocol. Okay. You need a roadmap. You need a plan to do a clinical trial. So therefore, you will have a protocol. And what are the elements of a protocol? When you're writing the protocol, these are the things that a protocol should have. It should have objectives. So objectives is, why am I going to do this study? Okay. My objective is to improve the outcomes of cervical cancer. Okay. So today, the survival of patients with stage 3 or 4 cervical cancer is, let us say, 50% at 5 years, let us say. So I want to increase it to 80% at 5 years. So that becomes an objective. Okay. To do the study, you need to then explain the background. So in the background, what do you say? Okay, if there is a stage 3 cervical cancer, what are the different treatments that are available? Okay, and what are the efficacy of each of these treatments? How long do people survive? And what are the limitations of those treatments? Whether there are any toxicities that are documented, and so on and so forth. And therefore, you say that there is an unmet need for new drugs, and therefore you say that there is this new drug. Then you write about the new intervention. Okay, this new intervention has so many advantages, and it is been shown to be very effective in earlier clinical trials. And therefore, I want to do a confirmatory phase clinical trial patients with stage 3 or 4 cervical cancer. So that is how you write the background and rationale. Okay, then you will have to mention about what are the inclusion criteria, what is the exclusion criteria. So all these things will come. So that these kind of things will come in the methodology. Inclusion criteria, exclusion criteria, what is your treatment plan, what are the study procedures, they're all methodology. For response evaluation criteria, maybe you'll be going to use RESIST criteria for response evaluation. You'll be using CTCAA criteria for toxicity evaluation, and so on and so forth. And you have to write the statistical analysis plan, which means how are you going to analyze the data, and you have to write a section on the sample size. Okay. And now I just want you to recall back the PICO that I mentioned above. So PICO I mentioned, no? P for patients. Okay. The P for patients is taken care in the subject selection criteria. Okay. And the treatment that you're going to deliver is intervention. I for intervention, that is nothing but the treatment. Then you have C for comparator. That once again comes in the treatment plan, because the treatment, suppose you are doing a randomized trial, you'll have existing treatment, which is a standard of care, and the new treatment. So both the I and C, intervention and comparator, both of them will be written in the treatment plan. And O is the outcome. So for outcome, you have response evaluation criteria. So that way, all those 4 aspects that are really pivotal for a clinical trial will be kind of elaborated in the protocol. Okay, under these headings. Next slide. Okay. Next slide. Okay, so now developing your question. So see, one of the important things that we said is, when you're writing your research plan, you should have your objectives very clearly stated, and you should have background and a rationale. So these 2 sections, will constitute developing your research question. So therefore, you should have a very clear purpose to start your study. So you must clearly know, okay, so I have a feeling that the outcomes of certain disease can be improved. Okay, and I have a feeling that there is this drug, because it has shown certain activity in previous studies, it can do the job for me. So therefore, I want to kind of use that for this given indication. So you state your purpose. Now, you already know the literature, okay, but you will also do more literature search, because at any given point in time, your knowledge may not necessarily be very current. So therefore, before you start writing a clinical trial protocol, it's important that you go back and do a complete survey of the existing literature to see what is available knowledge about the disease that you're trying to address, and what is available knowledge about the intervention that you're going to address. And then be iterative. Iterative means, it is possible that, in one go, you cannot get everything right. So a protocol is actually iterative, because there is a lot of movement back and forth. So you write it, you send it to your senior, he will make some corrections, he will give some ideas, he'll come back to you, he'll prepare the second draft, maybe it will go back to 3-4 other collaborators, it will come back with their comments. So that is the iterative part. Okay, and try to specify who, what, where, when, and your purpose. So that is the whole purpose of having that PICO and everything. Okay, so who is what type of patients, what type of intervention, what is the outcome of interest, and so on and so forth. And before, because clinical trial it takes a lot of effort. So to do any clinical study or research, it takes a lot of effort. So therefore, you have to be very, very certain that at the end of this clinical trial, you are able to add to the existing literature. So you must ask that question to yourself, okay, I'm doing so much of work, but will it make any difference to the people who are suffering from this disease? Will I be able to kind of improve it? Or will I be able to kind of disprove something? What is generally believed? Not everything that is believed, has to be correct. So somebody may be having wrong notions about certain treatment, which is a little unfounded. They may just be believing that it works without actually there being evidence. So at least can I disprove somebody that this what your approach is not correct, and therefore you should change your approach. So whatever. So you must always ask the question that what would the answer to this question add to the literature? Okay, will it make any difference? And if you're convinced that it makes a difference, you should go to the next level. Next slide, please. So you should go to the research process. Next slide. Next slide. Okay. So the research process is you have identified. So far you've identified the problem. You've done a literature search. You have raised the hypothesis. That okay, if I give this, the outcomes will improve. There is a hypothesis, and then comes your design. So how do you want to design? So you must always remember that it is the research question that drives the design and not the other way around. As I said, you cannot say that I know this design very well, so I'm going to address all questions through this design. No. Whatever is the question, the design should be according to that. And you must set in place a plan for data collection, because as I said in the first, data collection is one of the important aspects of good clinical practice, which will ensure the integrity of your research. So you must have a plan for data collection. So how frequently you want to call the patient for follow-up? How are you going to ensure the patient comes for follow-up? Are you going to kind of call the patient? So okay, saying, sir, your next visit is 1 week from now. So please make arrangements to come to the hospital. Okay. And once the patient comes, is there somebody who will actually talk to the patient and then, get the desired information? Like, for example, is there somebody who will say, okay, you need to do your toxicity evaluation. We need to do your response evaluation. Please go and get a CT scan done. And once the CT scan is done, somebody should kind of collect the information and enter it. Okay, what is it? Is the patient is in stable disease or a progressive disease or a partial response or a complete response, whatever it is. So once all the data is collected, there must be a statistician who is involved in the study who is able to do the analysis of the data. And one important thing I want to mention here at this point in time is it's always important to have a statistician involved in a clinical trial right from day one. There are many instances where you go to a statistician after you've collected all the data for a clinical trial, and the statistician is helpless, because sometimes he'll feel that if he were consulted before, he would have asked you to kind of collect the data a little differently, maybe at little different time points, maybe a little more extensively, or he would have advised that okay, the sample size that you did the study with is very few. Ideally, you should have had 300 patients instead of 200 patients that you have enrolled. So those kind of things. So therefore, always involve a statistician as soon as you start thinking about doing a clinical trial. And once the statistical analysis is done, then comes the interpretation of findings. So this is the research process. Next slide. And I already informed you that whenever you're doing a clinical trial, so the first step in a research process is you should have an informed consent form, because you have to tell the patients what are the aspects of the study. You have to give them information about the study. You have to give them information about what are the alternative treatments they get if they don't agree to participate in the study, and what are the problems associated with the drug, how frequently they have to come, what are the costs involved. All those information has to be mentioned in the informed consent form. Next slide. And all of us know, next slide, that you cannot start a clinical trial without IRB approval. So with all the protocol, informed consent form, and the data collection form, which is also called as the case report form, CRF. So you have to submit all these documents, the protocol, the informed consent form, and the case record form. All of them should be submitted to the institutional review board, and you should get their approval. Next slide. And then you will start enrolling patients. And the next slide, please. And what are the problems? So there are always problems with patient enrollment that you should be aware of. Like, for example, it is generally known that why trials do not work out is because of poor patient enrollment. When you're writing the protocol, you say, oh, our center is so big, we see thousands of cases, so I'll be able to do this 200 case study in 6 months. Okay. But then when you assume, when you're doing it, you will realize that, okay, some patients are not agreeing to take part in the study. Some patients are getting lost to follow up. and some patients are not getting referred to you when when you thought that they would be referred, and they're just coming to the OPD and somebody else is seeing and going away. So you're not able to kind of catch them. So on an average, not more than 5 to 6% of patients actually can be enrolled on a clinical trial. So you must be aware of those things. And based on that, you must make projections. And if you're not able to, if you think that you will, as a single center, you will not be able to complete the entire recruitment in a stipulated time, you should also make provision to have multiple centers in a study so that you can do a multi-center study and complete the study in proper time. Okay. Next slide. Okay. So therefore, you must follow certain recruitment strategy like building trust with your colleagues, and you have make sure that your staff, study staff, like your coordinator, contacts everybody in the hospital and keep them informed that you kind of put these printouts about the inclusion criteria, exclusion criteria in the OPDs, in all the OPDs in your hospital, so that people know that there is a study going on. Okay, and depending on the nature of the study, if it is a cohort study or a cluster randomized study, you can speak to community groups and then say that, okay, there is this vaccine study going on, so make sure that you enroll your community people into the study. And then sometimes, depending on, once again, the type of the study, newspaper and radio ads can also be given. You can post about your study on the internet, and physician referral. All these things can actually make your study enrollment faster. Next slide. Next slide. And when it comes to data collection, I told you about the case report form. Case report form is a structured way of collecting your data. Okay, visit one. What are the activities for visit one? And I'm going to capture all those activities in the case report form. Like, for example, if the visit one is safety evaluation based on physical examination. So then when the patient comes, the doctor would have examined, okay, the patient is doing well, he doesn't have any complaints, his abdomen is normal, chest is normal, heart rate is so much, blood pressure is so much, and all. So all those things, the same information, whatever has been captured, you should have has been written in the source. You should have provision to kind of capture in a case report form. So that is how you design your case report form. So that visit by visit, whatever information is important and relevant for the analysis of outcomes are actually captured in the case report form. Okay. And so the place where form, and there is somebody like a quality control person, a clinical science associate or somebody who will come and verify the data entered in the case report form with the source document, and he ensures that, okay, the data is captured adequately or accurately. So that kind of a two-step process also many trials follow, so that your data is more reliable and more accurate. Okay, next slide. And in fact, we can talk to you when we come there. We can talk to you about how to kind of design a case report form so that you get the information that you are looking for. Okay. Next slide. Okay. So now clinical trial end product. So at the end of all these exercises, okay, so you should have an outcome which is unambiguous conclusion regarding the clinical outcome of the test or device. So you may say that the test statement is useful, or you may conclude that the test statement is useless, is not good. But at least whatever you conclude should be very without any ambiguity, without any confusion. Okay, the trial may be a positive trial because you could demonstrate that your new intervention is better than the old one, or it could be a negative trial because you demonstrate that the new intervention is not as good as the old one. That is fine. But whatever you conclude, you should be able to conclude without any ambiguity, without any confusion, or without any doubt. Okay, and strive for an ideal situation. Okay, try to do things under ideal circumstances. But we all know that ideal doesn't happen in the real world, because there are always patients who will kind of be lost to follow up. They will not follow up with you for the duration of the trial, or your enrollment will not happen as you thought. you thought you'll be able to enroll 10 patients per month, but you're able to enroll only 4 patients per month. So all those problems will be there, but always try to make, have an ideal situation. But if it is not ideal, you should settle for the best compromise. But in the effort to do something the best possible way, okay, it is not like, okay, if I cannot do the best, I will not do anything at all. That should not be the attitude. So as I say, that the best should not be the enemy of good. Okay, if you cannot do best, try to achieve the next possible level, which is good. But it is not as though if you cannot do it the ideal way, you should not do it at all, because then you will never be able to make any difference. Okay, if you don't do it. Okay, so that is about the first section of the study about clinical research methods, where we discussed about what is a clinical trial, what are the different phases of clinical trial, and before that we discussed about what is clinical research, what are the two different broad types of clinical research. One is observational studies and intervention studies. Under the observational studies, we discussed case control study, cohort study, and cross-section study. And we understood that cross-section study is like a snapshot, taking a picture. Case control study is a retrospective study mostly, and cohort study is mostly prospective. Okay, and we also understood that there are 4 different phases of clinical trials: 1, 2, 3, 4. And we also understood that clinical trials can be done for anything. It could be a drug, it could be a behavioral intervention, it could be a diagnostic test, it could be a screening tool, it could be a surgical procedure. It can be anything. Intervention can be of anything. And we also understood what are the elements of a protocol, and having a protocol is very important. We discussed that how to do a literature survey, the importance of literature survey, so that you know everything about the current state of knowledge with respect to a particular condition and with respect to intervention. And then we also understood that what are the operational aspects of a clinical trial, like for example, in terms of drafting an informed consent form, having a case report form, having IRB clearance, and strategies for enrollment, and data collection, and so that the final end product that you get is actually accurate and reliable, and you should be able to conclude whatever you do with reasonable confidence. So that brings me to the end of the first set of talk, which is called as clinical research methodology. Okay, and I'll be now going to the next part of the talk, which is about prospective observational studies. But in the meantime, if you have any questions, you can ask me and I'll be able to answer. And or if you think that you need a 5 or 10 minutes break, we can always come back after the break and we can reassemble. So whatever you suggest. Hello?
Thank you, sir. Would you like to continue the second lecture at the same time, or you want to take some question and answer?
Yeah, I'll be happy to take question and answers, because otherwise it becomes very monotonous. So let us have some question and answers.
So for the participants, if you have any questions, I don't find any question in the chat box. So I think probably, sir, we can continue the second lectures, and by this time the participants can write down their question in the chat box, and we can discuss in the later part of the lecture.
Okay, but was it? I mean, do you think the participants could follow what I was talking? I mean, was it clear?
So this is a very tough question, sir. In the virtual program, it is the main problem, because we cannot see the faces of the participants. So we hope that everybody can enjoy this sessions.
But at least, do you think that the flow was okay? And
yeah, everything is okay, sir. I for myself, I think it is okay.
Okay, fine. All right. So let's go to the next part of the talk, which is about retrospective observational studies. So just to recap once again, we understood that observational study is the type of clinical research, and observational study has 3 components: cross-sectional study, cohort study prospective, and cohort study retrospective. And today I'm going to just talk about the retrospective observational studies. Yeah. So next slide. Retrospective studies, you must understand what is a cohort, because in most of the observational studies we talk in terms of cohort. So cohort is a group of individuals having a certain characteristics. Okay, it could be a cohort of people born in the year 2000. Okay, or a cohort of people who got married in a certain decade. Okay, or cohort of people who are smokers. Okay, or cohort of people who are exposed to radiation. So basically a cohort is a group of people who have certain traits or characteristics which are common to them. Okay, next slide. So types of cohort studies can be either prospective or retrospective. And as I told you that a prospective is when you kind of find out that there is one set of people who have an exposure or a risk factor, let us say, and another set of people who do not have a risk factor, and you are going to follow both the sets of people, those with risk factor, those without risk factor, and then you are going to see whether there is the outcome happens in a certain period of time. Okay, like for example, it could be diabetics versus non-diabetics and how long they live. Okay, your hypothesis could be diabetics have shorter lifespan as compared to non-diabetics, let us say. So then you have one diabetics, non-diabetic, you just follow them up and then see when they die. Okay, so that is one very simple example of a prospective cohort study. On the other hand, the retrospective study is we know the outcome. Okay, here in the prospective study, we know the risk factor. Okay, so the risk factor could be smoker versus non-smoker, diabetic versus non-diabetic. Okay, or somebody exposed to radiation versus not exposed to radiation. So that we know that, and then we follow them up and then see whether they will develop the event of interest. But in retrospective study, we have people who already have the event of interest. Like for example, somebody who developed cancer, lung cancer, and others who are not developed lung cancer. And then you will go back and see whether what is the prevalence of asbestos exposure in those with lung cancer, what is the prevalence of asbestos exposure in those without lung cancer. You understood, no? You go back in time. So that is a retrospective cohort study. Okay. So basically, if you want to kind of tell you in simple language the difference between a prospective cohort study and a retrospective cohort study. In a prospective cohort study, the outcome would not have happened at the time of initiating the study. Okay, you will have only people with and without exposure. Outcome is going to happen later in time. But in a retrospective study, the outcome of interest should have already happened. Okay, the outcome of interest should have already happened, and then you go back and see whether they had the risk factor or not. Okay, that is a major difference. Okay. So I think prospective cohort studies I will not discuss much because that will be discussed in the next class in greater detail. Okay, but just to tell you that in a prospective, just I'll give you this slide alone. So in a prospective clinical study, the sample is defined prospectively during or before exposure and before outcome occurrence. So you will define the cohort or the sample, prospectively. Okay, so you will identify people who are kind of at risk and those who are not having the risk factor. Okay, much before the outcome has happened. Okay, just to give you an example, the effect of working for pay on adolescent tobacco use. There was a publication in the American Journal of Public Health about the effect of working for pay on adolescent tobacco use. So what was the hypothesis here is, the hypothesis was adolescents who work for pay, okay, are more likely to develop tobacco habit as compared to those who do not work. Adolescents who work versus adolescents who do not work, if you compare. So the hypothesis here was adolescents who go to work, okay, have a higher risk of developing tobacco habit. So the cohort was high school students from Baltimore, Maryland. Exposure was working for pay, and the outcome was initiation of tobacco use. And the results was adolescents who work for pay have a higher risk of initiating tobacco use. You understood, no? So here what has happened is the initiation of tobacco use has not happened. What do they know is that there are certain section of people who are working for pay and certain section of students who are not working. Okay, they're only going to school and coming back, but the other people are working. And then they are followed up and see how many of people in each group become tobacco, initiate tobacco use. Okay, and they observed that adolescents who work for pay have a higher risk of initiating tobacco use. So like that, there are several examples, like for example, smoking and lung cancer. So the hypothesis is that the smoking increases the risk of lung cancer. They found that smoking increases the risk of lung cancer. Likewise, association between asbestos and lung cancer, very similar example. Okay, and finally, elderly vaccination and whether it can reduce the pneumococcal vaccination and whether it can reduce the risk of hospitalization from pneumonia or influenza. So once again, so you have a vaccination card, you have a non-vaccination card, you follow them up, and then you see what is the risk of hospitalization due to pneumonia or influenza. And that will give you the answer whether vaccination helps or it doesn't help. Okay, so that is about prospective studies. I gave you a few examples. Now coming to the retrospective cohort studies. Okay, so retrospective cohort studies means that the both exposure and disease have occurred at the start. Even before you start the clinical trial, both the exposure and disease have already occurred.
Sir, please instruct me to change my slides, please.
Sorry?
Please instruct me to change my slides.
Yeah. Please go to the next slide. Next. Come up to retrospective cohort study. Next. Yeah. So this is our slide, section of interest, because my job is to talk about the retrospective cohort studies now. Okay. As I said, both exposure and outcome has already happened. Okay. Data has been already collected because in the hospital, the people were coming, they had the exposure, they had the outcomes, data has already been collected. You just go backwards, collect the data, and then see whether what is the prevalence of the risk. I'll just explain that one a little better. Okay, just to give you one example, there was a study which was conducted in the year 2000. Okay, in patients who were surviving myocardial infarction. So they had myocardial infarction between 1986 to 1996, and they survived. And this study was happening in those patients. And then the outcome of interest was cardiovascular events 6 months after MI. Okay, and the exposed group was, so the hypothesis was, if you take lipid lowering drugs, you can prevent a second cardiovascular event 6 months from the time of MI. Okay, that was the hypothesis. Okay, so what is the exposed group? There's a group which was taking lipid lowering drugs like statins. There was a cohort which was not taking lipid lowering drugs such as statin. You follow, and then you already have patients who had a second cardiovascular event. Okay, then if you kind of calculate what is the prevalence of statin use in those who had a cardiovascular event versus those who did not have a cardiovascular event, then you'll be able to tell whether there is any benefit of taking statins or not. Okay, so that led to this publication, lipid lowering drug use and cardiovascular events after myocardial infarction. Okay, next slide. Next slide. Next slide, please. Yeah. Okay. So now just remember that example, whether use of lipid lowering drugs will reduce the cardiovascular events in patients who have had MI in the last 6 months. Okay. So now let me explain the design here. Okay. So you have cases. So cases are those patients who have cardiovascular event. Okay. Controls are those patients who do not have cardiovascular event. And in these cases, you have some section of them who had taken statins and some section of them who had not taken statins. And within the controls also, you have one section which has taken statin and one section which is not taken statin. That is exposure absent. So now you construct what is called as a 2x2 table. So you have disease present, disease absent. Okay, disease present, disease absent, and exposed and non-exposed. So exposed is those who are on statins, non-exposed is those who are not on statins. Disease is those who had a cardiovascular event after MI within 6 months after having an MI, Non-disease is those who did not develop a cardiovascular disease within 6 months after having an MI. And now you will fill this A, B, C, D. So A is this one, those who had disease and who took statins. So disease and who took statins. And then C is disease not exposed. So that is the second group. Then you have, they did not have disease, but they took statin. And they did not, the D is, they did not have disease and they did not take statin. So with this, you will complete what is called as this 2x2 table. Okay. So when you calculate the relative risk and when you calculate the attributable risk, when you calculate the incidence rate, for all those calculations, this 2x2 table will be very important, and that you will learn in the next few slides. Okay. So now, so when you do, why do you do a case control study? You want to do a case control study because you want to prove a hypothesis, and you will prove the hypothesis by calculating some of these basic measures, like what is the cumulative incidence, what is the incidence rate, what is the relative risk, what is the attributable risk. So cumulative incidence and incidence rate are measures of disease occurrence. Okay. So how frequently the disease is occurring. So that is given by the cumulative incidence rate. Okay. And measures of association is given by relative risk, because it will tell you what is the risk of disease happening in the exposed versus what is the risk of disease happening in the non-exposed. So you will get a relative risk, because what is the risk in the exposed group relative to the non-exposed group. So that is called as a relative risk. And attributable risk is a slightly difficult concept. So therefore, I'll explain it when I come there. Okay. So what is the cumulative incidence? The cumulative incidence is the risk of developing disease. Okay. New cases of disease versus divided by the person's at risk. I'll just explain what I mean by that. So just understand. Go to the next slide, please. Next slide. Yeah. So I explained this one. I explained about the measures of disease occurrence, which is cumulative incidence and incidence rate, and measures of association. So there we talk about relative risk and attributable risk. The relative risk is what is the risk of disease happening in the exposed versus the disease happening in the unexposed. Okay. So that will tell you how many times more risk is there in the exposed versus the unexposed. Next slide, please. Next slide. Okay. So now cumulative incidence. So what is cumulative incidence? You just go on adding. Like for example, okay, 1% had an event, 2% had an event, 3 had an event. So that is the cumulative incidence. And you divide that by the number of people who are at risk. Like for example, next slide. Next slide. It becomes very clear here. Okay. So you're able to see the 2x2 confusion matrix, also called as the 2x2 table. Okay. So what is the risk of disease in the exposed? Exposed, total number of exposed is A plus B. The number of people having the disease is A. Okay. The risk of disease in exposed is A upon A plus B. Similarly, the risk of disease in non-exposed is C upon C plus D. So this is a simple incidence. If I ask you, what is the incidence of the disease in the exposed? Your answer should be A upon A plus B. If I ask you, what is the incidence of the disease in the non-exposed? Incidence of disease in non-exposed, C upon C plus D. C plus D together is the non-exposed, that is the denominator, and C is the patient with disease. So therefore, C divided by C plus D is the incidence of the disease in the non-exposed. Next slide. Next slide. Okay, now incidence rate. So incidence rate is a slightly different concept than risk than cumulative risk. Cumulative risk is just telling you, okay, how many people got the disease out of how many people. Okay, like it is telling a percentage. What is your percentage exam? 80%, which means I scored 80 out of 100. Cumulative risk is something like this. Okay, but the incidence rate is if I observe 100 people, okay, for various periods of time, how many will develop the disease per unit time? Okay, I'll make that very clear in the next next slide. Go to the next slide, please. So this is called as the incidence rate calculation. The incidence rate calculation is somewhat done like this. Suppose you have 5 patients, okay? Just let's take 5 patients only. And here in the second column, you have number of years of follow-up. 2.1 years, 4.8 years, 3.2 years, 9 years, 7.2 years. And in the third column, we have event, whether it happened or not. So in the first patient, the event happened. Whatever the event was, it happened. Second patient, the patient died before the event happened. Because if the event was something like cardiovascular event, okay? So if the patient dies before, dies of, let us say, COPD before that, then this does not constitute an event, no? So like that way. So the third patient also died of, or let's say, road accident. He died of road accident before the event happened, okay? But he was followed up for 3.2 years. The fourth patient, we reached the end of follow-up, but he did not develop the event. The fifth patient developed the event after 7.2 years. So now, how much of time we have followed up the all the patients together? All the patients together we have followed up for 2.1 plus 4.8 plus 3.2 plus 9 plus 7.2. That comes to 26.3 years. And what is the incidence rate in these 5 patients? Only 2, because the first patient had an event, the second patient had an event. Nobody else had the event. Okay. So therefore, what is the incidence rate? So 2 incidents in 26.3 years. Okay. So 2 people had the event in 26.3 years. So therefore, the incidence rate is 2 events upon 26.3 years. So therefore, 0.076 per year. Okay. So this 26.3 is not from a single person. So therefore, it is a cumulative of all the 5 people. No. So that's why you say 26.3 person years. But actually, if you look at the cumulative, the incidence rate, it comes to 2 upon 26.3, which is nothing but 0.076 per year. Or 76 cases per 1,000 patients per year. So you can represent the incidence rate either like this. We can simply say 0.076 per year, or you can say 76. So 0.076 is nothing but 76 by 1,000. No? So 76 cases for every 1,000 patients per year. So that's the incidence rate. So you understand, no? You understood the difference between a cumulative incidence and the incidence rate. Cumulative incidence is how many patients were there and how many developed an event. That's all, simple. 2 out of upon 60 or 5 upon 90. That is the cumulative incidence. But the incidence rate is how many events happened over how many total duration of follow-up was there. And that will give you the rate. Rate will always have time in the denominator. You must understand that. So therefore, you have 0.076 cases per year. Okay, that is your rate. Next slide, please. So now you understood how do you calculate the cumulative incidence. But if I ask you what is the relative risk, the relative risk is nothing but the ratio of the cumulative incidence in the exposed and the non-exposed. Okay. So if the exposed has a cumulative incidence of A upon A plus B, and the non-exposed has a cumulative incidence of C upon C plus D, the relative risk is given by cumulative incidence in the exposed divided by cumulative incidence in the non-exposed, which is given here, A upon A plus B whole divided by C upon C plus D. Next slide, please. Okay. So now, why is this relative risk important? You may ask, what is the importance of why should I calculate the relative risk? So we all know that relative risk is the ratio between the 2 groups. Okay. You have the ratio of the cumulative incidence in the exposed group and cumulative incidence in the non-exposed group. Now, I'll ask you, if the cumulative incidence is the same in both the exposed and the non-exposed, your relative risk will be 1, which means there is no association. The exposure is not causing a higher risk of the disease. But if the relative risk is more than 1, it means that the risk increases as the exposure increases. So exposure increases the risk of the disease. If the risk ratio is 2, it means that there is a 2-fold increase in the risk of disease in the exposed compared to non-exposed. If the relative risk is less than 1, it means that exposure actually decreases the risk. This can happen with, let's say, statin use and cardiovascular event. If you're using statin, the cardiovascular events will come down. So therefore, you will have a relative risk less than 1. Okay. If you have a relative risk of 0.7, you can say that there is a 30% decrease in risk. 0.7, how did you get 30%? It is 1 minus 0.7 expressed as a percentage. 1 minus 0.7 is 0.3. If you express 0.3 as a percentage, it will be 30%. So there is a 30% decrease in risk in patients who are exposed, people who are exposed versus non-exposed. So this is how you interpret the relative risk. So therefore, if the relative risk is very high, it means that if you are taking the, if you are getting exposed, you are increasing the chances of disease. If the relative risk is very low, it means that if you're getting exposed, the chances of developing the disease is very low. Next slide. So this is just one example wherein, this was a study which was conducted in UK, and they found that, patients who are suffering from pulmonary tuberculosis, they undergo sometimes several fluoroscopic examinations. So, and every fluoroscopic examination will lead to radiation exposure. So they wanted to see that whether patients who had that fluoroscopic who had frequent fluoroscopic examination with tuberculosis had higher risk of breast cancer. So what are the two groups? You have the group with breast cancer, who have the group without breast cancer, because that is the outcome of interest. And then you just go back and see how many of the patients with breast cancer had fluoroscopic examination, and how many with fluoroscopic breast cancer did not have fluoroscopic examination. Likewise, how many patients without breast cancer had fluoroscopic examination? How many patients without breast cancer had fluoroscopic examination? So if you do that, so you have the case, that is breast cancer, control, which is a patient without breast cancer, and then going back and see what was the exposure, what was the not exposure, how many were exposed, how many not exposed. Then you'll be able to complete the 2x2 table, and you'll be able to arrive at the number. So now, next slide, please. Next slide. So this 2x2 table is telling you exactly that you have breast cancer patients, you have patients without breast cancer, that is in the third column, and in the first column, exposed is those who had frequent fluoroscopic examination, non-exposed is those who did not have fluoroscopic examination frequently. And you must understand that, even within breast cancer, you'll have patients who are exposed and unexposed, and even without those without breast cancer, you have exposed and unexposed. And this is how you get the ABCD that we discussed now. And based on this, if you calculate the incidence rate, okay, so so incidence rate is 1.5 per 1,000 women years. So how do you know the incidence rate? I told you, how many events have happened divided by what is the total duration of follow-up of these women. So these 1,047 women in the exposed group were followed up for a total of 28,011 years. Okay. Okay. So then in the non-exposed group, what is the incidence rate? It is 15 out of 19,000 women years. Okay, so you will get something like 1.5 as the incidence rate in the exposed, and 0.8 as the incidence rate in the non-exposed. So your relative risk calculation is also very simple. Relative risk calculation is what is the risk of disease in exposed versus what is the risk of disease in non-exposed. Like 14, risk of disease in exposed is 41 upon 1047. The risk of disease in non-exposed is 15 upon 717. So that will give you a relative risk of 1.9. Okay, so what does it mean? Women who are exposed to fluoroscopies have 1.9 times the risk of breast cancer compared to those who are not exposed to fluoroscopy. So did you understand this 2x2 confusion matrix? Okay, so those who were exposed to fluoroscopy had 1.9 times the higher risk compared to those who are not exposed to fluoroscopy. Okay, and you'll also get this idea by the incidence rate. Like incidence rate is definitely higher in a woman with exposure, and incidence rate is lower in women without exposure. So now that brings us to the next measure in a quad study, which is called as the attributable risk. What is an attributable risk? So an attributable risk is nothing but the difference in the incidence rate in the exposed and the non-exposed. So attributable risk is equal to incidence rate in the exposed minus incidence rate in the non-exposed. So we know that in the exposed, the incidence rate was 1.5 per thousand. In the non-exposed, the incidence rate was 0.8. So the difference between the two is 1.5 minus 0.8. That is 0.7 per thousand women a year. Okay. So excess incidence rate of breast cancer among women exposed to fluoroscopies was 0.7 per thousand. So this is how you calculate the attributable risk, and this is how we conclude the attributable risk. So there was an excess incidence rate of breast cancer among women exposed to fluoroscopies by 0.7 per thousand women a year. So that will become the attributable risk. So attributable risk is nothing but the difference of incidence rate in the exposed and the non-exposed. Okay. And that will also give you an idea. So if it is a negative number, like for example, if the number of events in the incidence rate in the exposed is less than the incidence rate in the non-exposed, it means that your exposure is actually giving protection. But here, what is happening is the incidence rate is higher in the exposed and lower in the non-exposed, which means that the exposure is giving you more harm. It is increasing the risk of breast cancer. So that difference is called as an attributable risk. So that comes to the end of the various measures that are used in case control studies. Like we understood the relative risk, we understood the attributable risk, we understood the incidence rate, and how to calculate the attributable risk. We understood cumulative incidence. All these things we understood. Now, what are the advantages of a case control study? So, for any study of association or risk, case control study is very good because it will tell you whether an exposure will increase the risk of developing an event or it does not increase very easily. Okay, so useful, like we saw that in the case of fluoroscopy and breast cancer, and there are several similar examples. You can read any number of examples about case control studies. No, so basically, exposure effect relationship. If you want to study, case control studies are best. Okay, exposure outcome relationship, if you want to establish the association between exposure and outcome, the case control study is very good. During the reproductive years, that could be another exposure. And the third exposure could be something else. So you can evaluate multiple exposures and outcome associations in a single study. Okay, so and it is very quick and cheap to do. It doesn't take too many patients to complete study. Okay, and especially if the outcome is very, very rare. If it's a rare disease, you cannot do it in a prospective way, because if you do it prospectively, I told you, so case control studies are very easy to do. You will need very fewer patients, and it is a gold standard when you have to establish the association of risk and outcomes. It's the gold standard study, and you can also evaluate multiple outcomes. As I said, if you want to test the effect of contraceptive pills on breast cancer, and at the same time, in the same cohort, you want to answer exposure to fluoroscopy and risk of breast cancer, that can be easily done. And you can also study the interaction between contraception use and fluoroscopy. So those are several advantages. And especially if the outcome of interest is very rare, you cannot do a prospective study, because then you'll have to wait for forever to for the outcomes to occur. And because case control study starts with patients who are having the outcome. Okay. So confounders, like for example, age can be an important confounder when you're studying the risk of development of cancer, smoking and cancer. So you have to match the age and other various factors that may actually be responsible for the outcome by themselves. Okay, some of these confounders are they're the ones who will have an independent effect on the outcome, not related to the exposure itself. So if there are any such confounders, it's important that you balance those confounders between the two groups. Like for example, when you're doing an association study between smoking and lung cancer, it is important to balance the two cohorts for age. It may be important to balance the two cohorts for socioeconomic status, because they may independently have an effect on the outcome. Okay, And sometimes selection of control groups can be kind of little tricky if you're not designed the study properly. But more importantly, I think it's the recall bias and the confounders, which are the major disadvantages. Attention to those biases. Okay, next slide, please. Next slide. Next slide. So when to apply cohort case control design? So case control study basically. In many cases, case control studies are preferred to RCTs because they do not require strict randomization criteria. And sometimes if the RCT is unethical or improbable, like for example, I said in the earlier slide also, if the event is very, very rare, then you cannot do a prospective study. Okay. So under those circumstances, you have to do a case control study. And if you want to study the association between, let's say, asbestos exposure and cancer, you cannot do an interventional study. Okay, but I'll give asbestos to some people and I'll not give asbestos to some people, and then I'll follow them up and then see whether they develop cancer or not. That you cannot do. You have to do observation studies only, because those who are naturally exposed to asbestos versus those who are not exposed, there you can see a difference. So those things can be answered only through a quad study, but it cannot be answered through a prospective interventional study because it becomes unethical. So if it is unethical or improbable to do a prospective study, that is when case control studies are the best to answer some of the research questions. Okay. That brings me to the end. Next slide is the thank you slide. Okay. So that brings me to the end of this talk. Thank you very much for your attention, and I'll be happy to take any questions that you may have.
So thank you, Vikram sir. It was a very elaborate presentation. I have signed out several times, so I didn't see the questions in the chat box. Still, I am not finding any. So probably Dr. Masum, you have one question was there. So you can unmute yourself and ask the question directly to Professor Vikram sir. Dr. Masum, I have seen one question in the chat box by you. Masum, can you hear me?
Sir, there are 4 or 5 questions in the chat box, please.
Okay, so if you have, yeah, Kobir, unmute Masum.
Okay, sir. From now, any participants can unmute themselves, please.
Masum, you can unmute yourself.
Thank you very much. Thank you, Vikram Professor, for your nice elaborate presentation. I am Dr. Mohammad Masur. Basically, I am from preventive background. I have a question regarding case control study, and it's a very basic question, basic dilemma. So, in case of case control study, the comparison is between the healthy population and specific disease group population, or specific disease group with other specific type of disease group, or both are correct, sir?
Okay, so that is a good question. Like, for example, everything depends on the question that you are asking, and it is very important. Like, for example, if you are in a hospital setting, let us say, and you are trying to see whether, vaccination, let us say, against pneumococcus will actually, okay. So under those, to take patients who are coming to your hospital as cases and controls. So the cases obviously is very simple. Those who require hospitalization for pneumonia, but the controls should be those who are kind of coming to hospital for other reasons, and you should take them as control. But if on the other hand, so there, maybe patients with other diseases will become your controls, but of course they should be matched for other risk factors like age and whatever else is important, for the occurrence of pneumococcal pneumonia. But on the other hand, if you're doing the studies in a community, maybe, it will be, you will be taking those, the people without disease will be actually healthy individuals. Okay. So I think, it depends on the setting where you are doing the study. and the type of question that you are going to answer. So both controls may be applicable, but but it depends on the situation. So that's why when I said, one of the pros and cons of case control study, I also mentioned that what is the control arm that you are choosing. So therefore, the choice of the control arm should be very, very important. If you take patients, like for example, in vaccination cohort and the pneumonia, if you take patients who are not coming to the hospital, let's say, so then, that may itself create a bias, because, the very fact that somebody is coming to the hospital may increase the risk of pneumonia. So which will be eliminated if you're taking general population. So that way, those factors have to be definitely considered, but it is a very, very good question.
Thank you, sir. Thank you very much.
COVID, can you please read out the questions in the chat box?
Sure, sir. There is a question from Dr. Altaf Hussain, and the question is regarding the types of clinical trial. It may be diagnostic, screening, treatment. So what could be the phase 1, 2, 3 in a diagnostic clinical trial?
Beautiful. Very, very good question. So, okay, let me give you one example. Suppose you are, let us say, CT scan versus X-ray. Okay. So CT scan, so has been invented very recently, and you want to do a phase 1 clinical trial of the diagnostic test. Whether you, so that phase 1 clinical trial is whether you can use CT scan to capture images of the chest, so that you can diagnose cases of cancer. Okay, so that is the story. So now for that you need the feasibility is whether patients are even willing to kind of are able to undergo that scan completely or not. Because what it means is, the patients will have to kind of lie on a couch, they have to get into the gantry, they may feel claustrophobic, and there will be so many problems. So they may or may not, I mean, here the patients are kind of able to do that. But the feasibility assessment is whether patients are able to go through that or not. Okay, so just to give you another example of feasibility, like for example, you are doing, let us say, bronchoscopic biopsy, okay, of the lung. So now, whether it is, so what is the feasibility assessment when you are using bronchoscopic biopsy? Whether you can insert the bronchoscope, well enough, whether you will be able to biopsy the lesion, whether the piece, the amount of the chunk of the tissue that you are getting is sufficient to do proper staining and diagnosis. So all those things. So those kind of assessments will still be required in a phase 1 clinical setting, trial setting. You understood, no? So just like how for drugs you see whether it can be safely administered or up to what dose can it be safely administered. Here the feasibility for a diagnostic test is whether it will yield the outcome that you are looking at. I mean, in the sense, what is the outcome you are looking at? You are looking at a good chunk of tissue that can be easily subjected to histopathological examination. But now, if your biopsy is not able to deliver that chunk of tissue, it means that it is a failure in the first stage itself, phase 1 itself. So that kind of testing, of a diagnostic method happens in a phase 1 clinical trial. Whether it is able to, like, I'll give you one more example. Like, for example, you have a diagnostic test which can happen only in Japan, let's say, because it's a very high-end technology available only in Japan. So now, but we are doing the test, enrolling patients in all over the world, let us say. So the first important phase 1 study that we have to do is, if I ship the sample from India to Japan, will the sample be stable enough so that it can be subjected to the test? Okay, that logistics, all those things will have to be tested in a phase 1 kind of a setting. Okay, so that is why it is called as a phase 1 is called as a feasibility study. Okay, whether it is feasible to do, whether the patient will become claustrophobic as soon as they get into the CT scan gantry and run out of it. whether that is that going to happen, or are you going to get a good chunk of tissue from bronchoscopic biopsy, or whether you are able to send a sample successfully in a cold shipment to Japan so that it can be tested there. All these things, feasibility assessment will come in phase 1 clinical trial of a diagnostic test.
Thank you so much, sir. But
a very good question. I'm really happy that somebody asked that question. It's a very good question.
Sir, there's another question from Dr. Altaf Hussain, and the question is: as clinical trial may be diagnostic, so is a lab investigation is an intervention?
No. I mean, so as the clinical trial may be diagnostic. Yeah.
I'm repeating the question. As clinical trial may be diagnostic, so is a lab investigation is an intervention?
So the lab investigation for the purpose of the diagnosis is an intervention, correct? Like for example, that's what you're testing. No, I mean whether you can So basically, let us say there are two modalities of coming to a diagnosis. Okay, one is a serum-based test, a blood test, and the other one is a biopsy. Okay, so a typical example is, I can give you is in the space of oncology, let us say. So you have this EGFR, mutation analysis, which is done from the lung cancer biopsy. Suppose I have developed a liquid biopsy technique, and I say that I can just tell you whether the patient is EGFR positive or negative looking at the blood sample. I don't even need a biopsy of the lung tissue. So then, if you do that test, okay, you will take a blood sample to kind of run that test for EGFR mutation. That is an intervention. Okay, because that is the intervention that you're planning to test in that clinical study. Okay, so therefore that qualifies as an intervention.
Okay, sir, thank you so much, sir. There's another question from Dr. A.B. Mannan, and the question is: in retrospective observational study, what will be the minimum sample size?
Okay, so the sample size calculation is all based on what is the incidence of the disease and what is the difference that you want to, what is the relative risk that you are anticipating, that you are expecting. What is the relative risk that you are expecting? What is the power and what is the type 1 error that you want to, that alpha and beta errors. Okay, so all those things together will help you to calculate the sample size. So for sample size calculation in a retrospective study, you should know what is the anticipated relative risk, what is the power, and what is the type 1 error. So if you know these three things, you'll be able to calculate the sample size. It is just like calculating the sample size for any drug trial in a randomized study, wherein you know what is the alpha error, what is the beta error, what is the anticipated difference between the two treatments. Okay, so likewise here also, the relative risk, alpha error, beta error will help you to kind of calculate the sample size.
Okay, sir. Sir, we have another question. To see association case control study is a good one. Which measurement is more reliable to see association in case control study?
Very good, very good. So really, see, generally in case control studies, we use either relative risk or more specifically, we use odds ratio. See, I did not want to confuse you between, talking about odds ratio and relative risk, but there is a certain technical reason because of which another measure called odds ratio is actually used in case control studies. But you can use relative risk also. It's not that you cannot use. So basically, the best measure of association is either the odds ratio. Of course, I've not spoken to you about the odds ratio, but I can talk to you when I come there on 16th of November, or relative risk. That is the best measure. But other measures like attributable risk are also used. And it also depends on, for what reason, you want to use that measurement. Okay. So in some cases, relative risk may be more kind of it may be better to represent the data in terms of relative risk. In some cases, it may be better to show the attributable risk. But by and large, the relative risk or the odds ratio are the important parameters of association as far as a case control study is concerned. Okay. But I'm sorry, I did not touch upon odds ratio because I did not want to confuse you with too many things. But generally, for case control studies, odds ratio is the preferred basic measurement. And for a prospective quant study, relative risk is the basic measurement. And there's a very subtle difference between those two. I don't think you need to worry too much about that. For the purpose of this discussion, we can use odds ratio and relative risk interchangeably. So we can assume that both mean one and the same. There is no problem with that.
Thank you so much, sir, for your beautiful explanation. We have no more questions in the chat box.
So, Kobe, unmute Masum.
But I must say that, this audience...
every participant can unmute themselves. I hope so.
Masum, you unmute yourself. Yeah, Vikram.
Yes, sir. I have unmuted myself. Sir. Am I audible?
Yeah, you are audible. Anything to say, Masum, from your end?
No, sir.
Okay, Vikram. So I think we are the session, and I was really enjoying what we were listening. And I mean, things when we listen is very easy, but when we try to practice, we again face the challenge. I don't know why we forget every time. So we need to listen to it again and again, as you said. So Shuman, are you there?
Right.
Okay, Shuman, we can finish it if you think.
Yes, thank you. So I just want to repeat few sentences. So you have already received a format. So try to fill up that format. If you find any kind of difficulties, please contact with our two coordinators over there. They will try to help you. And the very next session will be on the same day, same time. And it will be presented by Dr. Jan Shastri. And after that, there will be another virtual lectures. And then the D-Day, it will be on the 16th of November before the conference. And we will be a physical session. You will get all the faculties over there. So we want your very active participations, because after the total sessions, we need to see some good quality proposals. And from the top proposals, our faculties will select, and the Oncology Club will give them a handsome amount to conduct the whole trials. So thank you for the active participations, and see you in next week. So thank you, Vikram sir. Thank you for your nice presentation. Thank you all.
And in fact, I must say that I was really encouraged by the kind of questions that I received from the audience. So I'm very confident that we'll have a very fruitful and interactive workshop. So thank you so much.
And prepare yourself. So when you are here in the physical mode, you will get more number of questions.
Showers of questions.
Yeah.
Credit goes to you, Vikram, because your lecture has instigated this question. So credit goes to you.
Thank you, but thank you also for warning me. So I'll be prepared.
A good preparation.
Thank you, sir.
No, but I'm sorry that the internet connection was not. There was a few issues with the internet, so I'm sorry about that.
Suman, I think we can give this recorded version to all the participants so that they can listen to it before the next lecture.
Absolutely.
Definitely, they will get by 1 or 2 days.
Okay.
So see you, Vikram. Bye.
Thank you. Bye. Good day, everyone.